We are in the middle of a pollination crisis. Pollination is a biological process that allows plants to reproduce. For many of the foods we rely on, fruits, vegetables, nuts, pollination is essential. Over 75% of the world's leading food crops depend, at least in part, on animal pollinators. Without them, yields drop, diversity shrinks, and entire agricultural systems become unstable. But globally, pollinators are declining. Bees, butterflies, beetles, and even some birds and bats, many of them are under threat. Habitat destruction, pesticide use, climate change, invasive species, and disease are all contributing to widespread population declines. In the U.S., beekeepers lost nearly half of their honeybee colonies last year alone. Wild pollinators, many of which are solitary or highly specialized, are disappearing even faster in some regions. This matters far beyond the insect world. These species support global food production worth hundreds of billions of dollars each year. They're key players in maintaining ecosystems and biodiversity. Their loss isn't just an ecological concern, it's an economic and humanitarian one. Among all these pollinators, honeybees have taken center stage because they're the most widely used and increasingly the most relied upon. Honeybees are commercially breedable. That means they can be raised in large numbers, transported between farms, and deployed on demand during critical pollination windows. They're generalist foragers, capable of pollinating a broad range of crops. They've also become the species we depend on to help compensate for the decline of wild pollinators. Honeybees operate as a superorganism. Tens of thousands of individuals form a single unit. There's no leader, no command center, and yet the colony manages to regulate itself, maintaining temperature, dividing labor, raising brood, responding to threats, all through coordinated behavior. This is swarm intelligence, collective decision-making that emerges from simple interactions among individuals. The colony decides when to forage, where to find food, when to grow, when to split and swarm, all without any single bee knowing the full picture. And despite decades of study, we still don't fully understand how these decisions are made. Understanding swarm intelligence is very important. These collective behaviors directly impact pollination. How many bees leave the hive, which flowers they choose, and how they respond to stress, all of it shapes crop yields and ecosystem health. But it goes deeper than that. The way bees solve problems together, without any central control, gives us clues about how complex systems work in nature. It can help us design smarter algorithms, build better robotics, and even rethink how humans organize and make decisions. Honeybee hives are a window into how intelligence can emerge from the collective. To truly understand swarm intelligence, we need a system that can directly observe, monitor, and record the detailed interactions happening inside the hive continuously. In the past, researchers developed creative methods to study social interactions among honeybees. One approach involved attaching tiny QR code tags to thousands of bees, placing them in glass-walled observation containers, and using automated camera systems to track their movements. These experiments produced valuable data on how bees share food and information, revealing the structure of their social networks. But the approach had clear limitations. Tagging each bee was labor-intensive. The studies could only run for a few days, and most importantly, the bees were removed from their natural colony environment. As a result, the complex interactions that drive real colony development remained largely unobserved. The honeybee queen sits at the center of the colony's life. Every worker bee in the hive hatches from an egg she lays, making her oviposition behavior a key driver of colony development. Oviposition behavior is the queen's selective laying of eggs into individual brood cells, regulating the colony's population growth and structure. This brood nest activity directly influences two most important indicators of colony strength: population size and worker bee health. Around her, a specialized group of worker bees known as the Queen's Court performs vital tasks, feeding her, grooming her, and helping guide her movement across the comb. The queen spends virtually her entire life inside the hive, rarely if ever venturing outside once she's mated. And yet, despite her importance, queens are notoriously difficult to study. Stationary cameras often fail to track her for long. The hive is densely packed and constantly shifting. The queen is visually similar in color and size to many workers, and she's frequently surrounded or even completely obscured by other bees. This is where robotics and AI come into play. Advances in computer vision, machine learning, and mechatronics are transforming how scientists study animal behavior. From tracking birds with GPS to monitoring insects with computer vision, automated systems can now gather data continuously and catch interactions humans would miss. In this spirit, an international team of researchers developed the Autonomous Robotic Observation and Behavioral Analysis System, a platform designed to monitor the entire hive's activity non-stop, with a particular focus on continuously tracking the queen bee. At its core, ARoBA is a robotic gantry that carries an entire honeybee colony. Two high-resolution cameras move along either side of the hive, scanning both comb faces in real time. This allows for close-range, uninterrupted observation of the colony's inner workings. The system operates with three distinct robotic movement modes: scanning, which surveys the entire colony systematically; tracking, which follows the queen bee's movements as she navigates the hive; and focal sights, which allows the cameras to dwell on specific points of interest, usually individual comb cells, to monitor their contents in detail. The system tracks three main elements: the queen bee, the worker bees, and the contents of individual comb cells. The queen is the top priority. The robot uses priority-based scheduling to ensure she's always tracked. When she's active or moving, the cameras and AI give her full attention. If she's stationary or laying eggs, the system shifts to secondary tasks like mapping cell contents or counting workers. This smart scheduling lets the system monitor the whole colony efficiently, without ever losing sight of its most important member. One of the biggest challenges in designing the robotic system was ensuring it didn't disrupt the bee's natural behavior. To observe the colony authentically, researchers carefully engineered around key concerns. First, vibration. Since bees use substrate-borne vibrations to communicate, the robot's motors were mechanically decoupled from the hive, with rubber dampers preventing disturbance. Next, lighting. Because bees are sensitive to visible and UV light, the system used infrared illumination for nighttime monitoring. Invisible to bees, infrared cameras enabled 24/7 video capture without disturbing circadian rhythms. Temperature control was also critical. The experiment took place in a climate-controlled lab, preventing external thermal fluctuations and insulating the hive from the robot's heat. For long-term experiments, systems for ventilation, supplemental feeding, and medication delivery supported colony health. Together, these measures allowed ARoBA to monitor the hive continuously without altering natural dynamics. The researchers carried out a 30-day observation of a full-sized honeybee colony inside a standard two-frame hive. During the continuous operation, the robotic platform captured over 100 million high-resolution images, all without human interference. From this massive stream of data, the researchers extracted what they called key behavioral metrics, short for KBM, quantifiable indicators of colony behavior, drawn from multiple biological scales. At the microscopic level, they captured things like egg presence in a cell or the queen's posture while laying. They followed the queen's movement paths and counted the number of eggs she laid each day. At an intermediate or mesoscopic scale, they analyzed how brood was spatially organized and how worker groups moved and clustered. And at the macroscopic level, they observed long-term trends, daily rhythms in queen activity, colony growth trends, and changes in brood development over time. With the multi-scale framework in place, researchers extracted detailed insights, starting with the queen. The robotic system tracked her continuously, building a layered profile. At the macroscopic level, it measured her comb coverage. Over 30 days, she walked more than 1.5 kilometers, reflecting steady patrols even late in the season. At the mesoscopic level, the first passage time is tracked, meaning the amount of time it takes for the queen bee to return to a previously visited location on the comb. Her first passage time is highly non-uniform and preferred the brood area. At the microscopic level, tracking the queen's position, orientation, and speed enabled the system to detect egg-laying behavior, characterized by stillness and abdomen insertion, allowing automated logging of approximately 187 eggs per day. Court bees feeding and grooming her were also monitored. While court size stayed stable, its composition shifted, highlighting her dynamic social network. Together, these observations formed the first continuous, non-invasive behavioral profile of a queen bee over weeks. The system also revealed how workers organized around the queen and brood. At the macroscopic level, worker density maps showed day-night activity cycles, with clustering near brood areas during active hours. At the mesoscopic level, interactions formed layered structures, a stable court close to the queen, surrounded by a fluid halo of transient workers. At the microscopic scale, workers reduced their speed significantly when approaching or being close to the queen, suggesting attentiveness, and stay durations varied, illustrating a nuanced social gradient. These patterns showed workers acting as a coordinated collective to support colony function. Beyond individuals, the system tracked comb development using deep learning. At the macroscopic level, it mapped centralized oviposition zones shaped by environmental and social cues. At the mesoscopic level, it measured stable brood compactness and resource rings. At the microscopic scale, it tracked cell development from egg to capped pupa, with most cells capped within 5 days, reflecting healthy growth. It also captured cooperative behaviors like workers preparing cells ahead of the queen's arrival. These insights transform the comb into a dynamic behavioral map, revealing the co-evolution of individual and collective processes inside the hive. This work presents the first fully autonomous, long-term system capable of continuously observing and analyzing honeybee colony behavior across multiple scales. The core contribution of the robotic system lies in its ability to capture and quantify the complex natural behaviors of the queen, worker bees, and brood, ranging from fine-grained individual actions to colony-wide dynamics, all without disrupting the hive. By combining robotics and machine learning, the system doesn't just track individuals. It extracts structured key behavioral metrics across spatial, temporal, and social dimensions, something previously unachievable with manual observations or short-term automated tools. That said, the system has limitations. Because it can't monitor everything at once, it relies on predictive scheduling, focusing on likely events such as queen oviposition. This prioritization means that low-frequency or unexpected behaviors may go unnoticed. Its performance also depends on visual clarity. The transparent hive cover must remain clean. Over time, dust, condensation, or substances deposited by the bees can degrade image quality and affect tracking accuracy. And while the system ran autonomously for 30 days, it wasn't immune to occasional breakdowns, highlighting the challenges of running delicate hardware in a biologically active environment. Looking forward, the potential is wide-reaching. One avenue is using the data collected to develop models to predict early warning signals for colony stress, disease, or queen failure. On the hardware side, future versions could incorporate localized lighting, thermal imaging, event-based sensors, or laser scanning to improve precision and expand what can be observed. More broadly, this project shows how robotics and AI can open new frontiers in behavioral ecology. It offers a scalable, objective, and reproducible way to study social animals over long timescales without intrusion. And while this study focused on honeybees, the approach could be adapted to other species and systems. Here are some final thoughts on why this work matters. This robot won't appear in commercial beekeeping anytime soon, and it won't solve the pollination crisis on its own. But as a research tool, it matters deeply. It represents a novel instrument, one that lets us observe and record what was previously impossible. Large-scale, multi-dimensional datasets that can reveal the hidden dynamics of honeybee colonies. This kind of data is critical to understanding colony behavior and development, which may ultimately support efforts to address pollination challenges and even drive breakthroughs in swarm intelligence. Throughout history, new instruments have been the foundation of scientific discovery. When Antonier van Leeuwenhoek first peered through his handmade microscope and saw bacteria in a drop of water, he entered a whole new world of microbiology and changed science forever. The impact of this robotic system is clearly not as revolutionary, but within the field of honeybee research, it marks a shift toward a data-driven, systems-level approach, one that could transform how we study and support these vital pollinators.